Hi, I'm Dr. Arvind Kumar, working as an associate professor, and my research background is like cancer stem cells and research lab, and we are focusing our research deals with the all cancer stem cells and also cancer cells. Being a dental college from Savita, we are expanding primary culture cells from the oral cavity like gingival fibroblast, gingival tissues and periodontal ligament tissues, and also dental pulpal tissues. Our main core is like we have to expand the primary cells from the oral cavities, and we have to check the biocompatibility and cytocompatibility of the dental materials. Okay, and being experienced in last 10 years related to the stem cells and cancer cells, and my research strength is like checking a new drug discovery and behind the mechanism of molecular mechanism begins in cells and stem cell related research and regenerative medicine also. So I'm happy to welcome you through this video to share my knowledge, to give a demo about the how to expand the primary cell culture and what is the applications we are dealing with the stem cells. Welcome you all. So as I said earlier, like we are expanding the primary cell cultures from the dental cavity, like periodontal ligament tissues and gingival fibroblasts and dental pulpal tissues. And we are the one we initiated and expand the primary culture cells in from the dental cavity, so oral cavities, and we check the biocompatibility and cytocompatibility, and say for example dentures and implants from PEEK and zirconium oxide, and also from titanium plates, and also we are checking the pockets sponges for hemostatic pocket sponges to check the biocompatibilities. And these are all the demos we have so far routinely doing the experiment. Before I enter the experiment, I want to show that the accessories with whatever we are using for the cell culture. Okay, so these are the dental materials accessories we are using for to check the cytocompatibility and biocompatibility. This is the basic routine consumables and accessories whichever we are using for the cell culture, and I am now going to give a live demo for a cell culture technique. So before we enter into the live practical demo about the culturing and isolation and culturing and expanding the stem cells, I just want to give a small introduction about the in vitro cell culture. And generally, why we are doing in vitro cell culture is like we want to check, say for example, we want to check any new invention drug or any new material to check their cytocompatibility or biocompatibility before entering into in vivo culture. So that we are introduce these techniques, and some experiments they were already listed, and then the standard operating procedures are already existing. We just following the SOPs to check the materials biocompatibility and check. As you all know about, our human body made up of blood, flesh and bones, but how many of the not aware about that whole body is arise from the single cells. So when I started my research career, I just wondering about how does a single cell become a body, human body. So behind on it, I started my research, and then we expanding like that the single cells is none other than it's a stem cells. So these stem cells are very unique, and they have a unique characteristics. They can be divided or they can be differentiated into any form of our body parts. So our main focus is how the stem cells differentiated in different form of our body parts or for different form of tissues or cells, and what is the mechanism behind on it. So in that being a dental domain, we only focusing on the dental pulpal tissues and dental pulpal stem cells, and check how it is helping helping for regenerative applications, especially in dental domain. Before we know about the in vitro cell culture, we should know about the basic fundamental and what are the equipments we are using for the in vitro cell culture. First one is for in vitro cell culture, the main important thing is CO2 incubator. Why it is very important is our human body have maintained same constant temperature and constant oxygen supply and constant carbon dioxide supply be there for inhalation exhalation. So that in vitro culture ideology is we have to mimic the human body that atmosphere into the culture room. So we have here that CO2 incubator having 37 degrees Celsius and 5% CO2 incubator CO2 supply and humidity about 92%. So and moisture content always supposed to be as like our human body conditions. So in this culture setup, in this CO2 incubator setup, we could be able to mimic our human body systems, that the body temperature and oxygen and CO2 supplies and etc. And here some cells are maintaining as for the experiment purpose. And apart from that, there is an inverted microscope to check the cell morphology before to monitoring whether the cells are healthy or unhealthy or whether it got contaminated or not contaminated. For that, this is a monitoring purpose to track how our body systems are monitoring with the help of that normal ECG or whatever that. Likewise, the cells also monitoring with the morphological studies. Apart from that, this one is inverted fluorescent microscope. From this, we could be able to see the live cells with the help of fluorescent dyes like Alexa Fluor, DAPI and Psi3. So usually that fluorescent microscope have three different color range. One is red and green and blue. With the help of three different dyes, we could be able to track the particular cells morphology and their internal organs, organelles and the nucleus also. Okay, and centrifuge also very important thing to separate and isolate and culturing and further subculturing the cell lines. Okay, so this working platform, this is the in vitro cell culture working platform, so called biosafety cabinet. Our biosafety cabinet is class 2 B2. So here the mechanism is that outside air should not enter inside, and the inside air should be circulated, and then it will go towards the outlet duct. The purpose of this is to avoid the contamination by blocking the outside air should not enter into inside. Okay. So now I am going to give the demo about how to isolate the gingival fibroblast from the gingival tissues. Usually we collect the samples from the volunteers, patients after the clearance of ethical and no objection from the patients. We collect the gingival tissues, and usually there are two different type of isolation is there. One is chemical enzymatic digestion, another one is mechanical digestion. It's nothing but that mechanical digestion is nothing but there is a chopping of small pieces of each tissues with the sharpened blades. Okay. Yes. At that enzymatic digestion is nothing but chopping the tissues. After that, we have to incubate with the collagenase type form for 30 minutes to dissociation of tissues as a single cells. And these enzymes will help to dissociate the cells become single cells from the tissues. So now I am going to give step by step demo how we are digesting the tissues with the help of sharpened blade, forceps and sharpened blades, and also for expanding the isolating and expanding the gingival fibroblasts. We need major one is a sharpened blade, and this one is petri dish, 10 mm dishes for chopping and then isolating the gingival tissues for culture expansion. This is micropipette to helping. It's help to collect the media or any solutions from the stock to our desired place. Okay. So before we start to expand or digest the tissues, we should wash with the one wash with the antibiotic containing normal saline. Okay, normal saline, and make sure that tissue is supposed to be perfused with the normal saline, and try to avoid as much as that blood stain or whatever the debris supposed to be discharged or separate from the tissues before we start the mechanical digestion of the tissues. So before we start the procedure, I would like to show the what are the chemicals and solutions we are using for the cell culture. So generally for the cell culture we have to use the cell culture media which contains all micronutrients. See here, like there are L-glutamate, sodium bicarbonate, liquids, sterile water, and suitable for it's supposed to be, it contains all micronutrients and endotoxin free and pathogen free, this one, and we purchase from the Sigma. And apart from that we are using phosphate buffer saline without calcium and magnesium. Why we are using without calcium and magnesium? It helps to detach the cells from the surface bottom visually. And cell culture, all items whatever we are using here, the media or any antibiotic solutions or phosphate buffer, all supposed to be cell culture grade. And whatever we are dealing inside the culture wood, it's not a matter. Our primary purpose of that doing all the procedures inside the wood is to try to avoid the contamination. And experience can help you to improve your skill of handling the culture cells and to avoid as much as contamination free cell cultures. And we are the primary source for the contaminations, because what are the materials and the cells are always inside the four walls, but we are the one we are accessing outside and then coming inside. So while entering inside and going outside, we supposed to be sterile with the help of IP 70% alcohol. It helps to sterile the pathogens, airborne pathogens or any microbes to kill, and we supposed to be in a sterile condition while accessing or dealing all the tissues and cells inside the culture room. And we should not touch any outside. And so here we collecting the gingival tissues from the human patient, and now I am going to digest with the mechanical method, chopping with the sharpened blade. So already I made one wash with normal saline, contains antibiotic solutions. That antibiotic solutions is nothing, it's a penicillin and streptomycin. After one wash, gentle wash, we discard the antibiotic containing saline solutions. Then we wash with media which we are going to maintain the tissues after the digestion. After washing with the media, we are going to discard the media without any disturbance of the tissues. Then immerse with the media for a while until we take off the tissues from the tubes for the enzyme mechanical digestion. In the middle of the experiment, frequently you wipe with the 70% alcohol to avoid the contamination. So now I washed the tissues with the saline contained antibiotic solution. Afterwards, I transfer the tissues with the micronutrient enriched media. Now the tissues are ready for the mechanical digestion. So now I am going to gently transfer the human gingival tissues into petri dish. Make sure that the tissues should not be air dry. So now I am going to add few drops of the media. Let it be immersed into the media, the tissues. As I said earlier that there are two types of digestion. One is mechanical, another one is enzymatic. Now I am doing mechanical digestion. It's nothing but like pinch of tissue has chopped with the sharpened blade with the fine pieces. So while doing the small pieces, so some tissues dissociate, become a single cell. Sometimes some bunch of cells become a colony. Try to chop it as much as fine pieces. See here, initially it was a pinch of tissues, now it will become fine pieces. With the help of sharpened blade, I chopped it in a fine pieces. So now we are half of the way in the experiment, middle, and now I am going to add micronutrient enriched media which contains glutamate, essential amino acids and salts, basic salts. It will help to retain the cells become live, and these media also contain 20% fetal bovine serum. So now that fine pieces is immersed in the micronutrient enriched media. Okay. So I immersed with the micronutrient enriched media with our interest of tissue collected from the gingival fibroblast, and now I'm going to cut expand the tissues in single cell isolation and expanding the human gingival fibroblast with the enzymatic digestion. So enzymatic digestion is nothing but incubate the enzyme, the fine chopped tissues with collagenase type 4. This will help to dissociate the cells from the tissues as a single cell. After incubating so now I am starting the preliminary step of the enzymatic digestions. This only slight changes would be there in enzymatic digestion when compared with the mechanical digestion. What we are doing is, like for a mechanical digestion, just we chop into small pieces, but in enzymatic digestion, after chop small pieces, we incubate the fine tissue pieces of these tissues with the enzyme collagenase type 4, which will help to dissociate the single cells from the tissues. So now I am making it small pieces. As a fine pieces with the help of the sharpened blade, surgical blade, we incubate the cells with collagenase type 4 enzyme for 30 minutes. Try to collect the leftover small pieces, stick as the side edges of the sharpened blade, which helps to yield more single cells from the tissues. So, now I am adding collagenase mixed condition media for enzymatic digestion, which helps to dissociate the cells from the tissues. So why we want to do this such a dissociation means the duration of single cell formation from the tissues, that they will be reduced when compared to mechanical digestion. That's why we suggest, if the sample and the study is very precious, go for enzymatic digestion rather than to fastening the experiment to accomplish. So after 30 minutes, we replace the media with the micronutrient enriched human gingival fibroblast condition media. So this is the preliminary step of how to culture the primary cells from the tissues in any source like human, animal model, say for example bone marrow from the mice, to isolate the MSC cells, mesenchymal stem cells from the bone marrow. Okay, so now I am going to put inside the incubator, and very next day we should monitor the dish. After the enzymatic digestion, I am going to place the tissue containing petri dish into the CO2 incubator, and very next day we have to monitor whether the media has discolorate or any contamination is there. If is there any no contamination or if there is no decoloration, we supposed to leave it without any disturbance for minimum 3 to 4 days and let it to attach the tissues in the bottom of the petri dish. After the attachment, the cells will start arise single cells from the tissues, and it will take minimum 1 week to 10 days. Okay. So okay. So after the cell has attached, tissue has attached at the bottom, after 10 days, the cells will start to arise as single cells from the tissues. Dense of the inner core is nothing but the fine chopped small pieces of tissues arise, and you could be able to see the surrounding places. There is a single cells as arise from the tissues. This is called after 10 days you could be able to see the single cell array formation arise from the tissues. Okay, so after some days the cells will get wide and spread, and you could be able to see monolayer cells arise from the tissues, and it start to spread all over the surface area of the empty space of the petri dish. So we leave it, the cells, let it be until it get to start to spread all over the space to cover the empty space. After that monolayer cells formation, we trypsinize the cells with the help of trypsin. It's an enzyme. It will help to detach the cells from the surface bottom for the further passages. Usually, the primary cultures, when it reaches the P2, then only you will get the monolayer cell culture, that homogeneity. Usually, the monolayer cell culture, that homogeneity will start from the P2 passage. Before that, there are bunch of different type of cells will be there, like fibroblasts, epithelial cells, and also some RBCs also will be there. But after a week, that second passage, you will get the homogeneity cells will be there, uniform morphology, and same cell, the majority of population cells will be it. 90% of the cells will be fibroblasts. Okay. Now I am going to give the small example for homogeneity of fibroblast cell. Here we taken up into P2 passage, P2. This is the ideal example for the human fibroblast with the uniform morphology. You could be able to see all the cells are in same morphology and also spindle shape morphology and same sizes. It's so called homogeneity. Homogeneity. Same called homogeneity. So here only cells are there. There is no contamination. There is no debris. There is no any bacterial contamination. If it is any bacteria means you could see this is the live imaging. You could be able to see any movement, motility is there. There is no motility. Only the cells are there. We are screening the live cells with the help of inverted microscope, phase contrast microscope. Whatever I showed you, phase contrast normal morphology microscope. Now I am going to do another one assay, which is live cell imaging with the help of acrylamide orange, which emit green fluorescent. So green fluorescent means whichever healthy and live cells will obtain or uptake the green dye with from and so we under the microscopy while observing, it will emit the green fluorescence. So it confirming that the cells are live. There is no any cell death. Now what I am doing is to check the cell viability with the help of acrylamide orange staining. So I am replacing media. Already I pre-seeded the cells last day itself into the 12L microplate, and now I am going to add fluorescent dyes, that acrylamide orange, nothing but acrylamide orange dye onto the cells and incubate for few minutes. So after the incubation for 4 minutes, I am taking off the dye and gently replace with phosphate buffer saline. So again one more wash to discard the leftover dye and with the help of phosphate buffer saline. After discard the fluorescent dye, now I am going to place it in inverted fluorescent microscope to observe the cells, whether it has uptake the dye or not, and with the help of fluorescent microscope. So I am now placing it into inverted fluorescent. So this is a phase contrast bright field. You could able to see the cells here. I could show, yeah, single cell. Yes, see here, these are cells, and you could able to see here there is a nucleus. There is a 2 dots. It's a nucleus. Okay, it's a bright field. Now we already added a green fluorescent dye, so called acrylamide orange dye. Now I am going to switch into green fluorescent lamp. So see here, the cells are uptake the green fluorescent dye and emitting the green fluorescent while absorbing under the green field. So, usually the technology the characteristics when the cells are emitting green color, it's confirming that all cells are healthy live cells. When it become yellow or red means it's early apoptotic or late apoptotic. It turns red color means it become necrotic cells, but all field showing green color. So we are confirming that all cells are healthy and normal. Okay. See here, the primary culture cells isolated from the dental pulpal tissue. This cells, and we add green fluorescent dye and observed under the green fluorescent, inverted phase contrast from fluorescent microscope. So in green, under the green fluorescent lamp, it emitting in the green color the cells. Okay. From this session, I shared my knowledge and working skill about the how cells isolate from the tissue, human tissue. Primary cells isolate from the human tissue for the further experiments and also for regenerative applications, also translation research. Our lab also dealing with some bioscaffold, biomaterial related studies also we are doing it, and this is our lab normal routine experiments and then research. So far we are doing here. Thank you.